Hello, everyone, and welcome to today's webinar, Scaling Up Exosomes and Other Extracellular Vesicles. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Beckman Coulter Life Sciences. To learn more, visit them at beckman.com. Now, we encourage you to participate today by submitting any questions you might have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing this presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain those credits. I'd now like to welcome our speaker, Dr. Derek Lenz, Senior Manager, Biopharma at Beckman Coulter Life Sciences. Dr. Lenz, welcome. You may now begin your presentation, sir.
Welcome, and thank you for joining us today. My name is Derek Lenz. I'm the Senior Marketing Manager for Biopharma at Beckman Coulter Life Sciences. What we would like to present to you today is scaling up and scaling out in extracellular vesicles and the use of centrifugation solutions in managing the quality controls of these processes. Before we get started, I'm obliged to show you the intellectual property disclaimer. This is solely owned by Beckman Coulter Incorporated, and any use, copyright, or distribution without consent is strictly. So just to introduce, the origins of the field of biopharma derive from the Division of Clinical Research in the U.S. Bureau of Medicines, but it's a little more complex than that because the authority vested for that is actually held by the Division of Biopharmaceutics in the United States Food and Drug Administration. So this is generally considered the onset of the field. But shortly thereafter, numerous bedrock concepts and parameters were developed and published over a range of topics that I cite here. And it's at this point where protocols for regulatory consideration and for the scale up and scale out of biologics also began. By extension, however, this is also the point where many of the challenges we face in scaling up and scaling out are rooted. So let's talk about some of the key objectives we'd like to cover today. We'll define the biopharma landscape, and looking at some scale up and scale out details, looking at the rapid evolution of this field. We'll then utilize exosomes as a model for the scale up and scale out of biologics. And then we're going to highlight, I think, a very seminal publication that really served well to advance the field of exosomes and other EV research, utilizing centrifugation as a means of really isolating pure cultures and scaling them up for material research downstream. We'll then summarize, conclude, and have a nice discussion in our Q&A. So as I mentioned, the field of biopharma is a rapid dynamic field that's been evolving very quickly. Since the 1980s, when we began research on genetically modified agricultural organisms, to here in 1990, where we initiated the Human Genome Project, and we had our first gene therapy experiment in humans. Not 7 years later, the FDA had started approving novel biologics in the treatment of disease indications, as I cite here. In 2000, the U.S. venture capital fund for biotech was estimated to be around $6 billion. And I find this very interesting. By 2002, Lipitor, one of the most successful drugs to ever be marketed, in this case manufactured by Pfizer, had annual sales reaching $8 billion. And then lastly, in a matter of 11 short years, from 1990 to 2001, we had our first draft sequence of the Human Genome Project published in Nature and Science. So a rapid and dynamically evolving field that we are in by pharma. So let's switch to this concept of scale up and scale out in biologics I use a very generic workflow here, a process where we have discovery, clinical trials, and then commercialization. Within those steps, there are a number of key aspects such as designing the potentially commercially viable biologic, screening it and finding it. Once we find it, to select it, develop it, tech transfer, where we ultimately want to produce, scale up, and certify. So looks a little simple on this slide, but obviously much more complex. Now let's use a simple example of cell cultures in a bioreactor where we will subsequently harvest, clarify, and isolate our biologic product of interest. When we're talking about these concepts and scaling options, the first available to us is scale out usually, right? Increasing our capacity to increase our yields. We may then decide to scale up to increase the throughput. So capacity increase and throughput increase to ultimately affect, hopefully in a positive manner, operational efficiencies if we've made the correct selections and we have run our protocols correctly. So here's some examples of that, right? If it's a bioreactor, again, we may increase the volume of the bioreactor to improve capacity, and when we hit our milestones, replicate those to increase scale up and throughput on the means to increasing and improving operational efficiency. Now, I think most of us that have worked any amount of time with biologics appreciate, while this may seem fairly straightforward on paper, in reality, it's rarely a linear scale game. There are a myriad of nuances and complexities in biology and in production of biologics. Small variations such as a material composition or bench technique can induce dynamic magnifying changes, and that variability really causes challenges to us collectively in the field. So just one simple example to highlight the complexity and the lack of linearity in scaling up biologics. I'd like to show the HEC-293 cells. More and more becoming popular because it grows quite well in culture. It's easily transduced. It takes on transfecting agents quite well, and it does produce, if you're particularly working with proteins, high quantities of proteins of interest. However, most of us just assume certain concentrations of cells generically, right? 2 times 10 to the 6th, 10 to the 7th. I think we've all kind of done that when we started working with biologics. Yet here, in an insert cited by the ATCC, it recommends keeping densities low, 1 to 3 times 10E5 per mil, concentrations of even 10E4 per mil when we're seeding. And in most cases, they recommend not to exceed 10E6 per mil. And I think the picture here on the right-hand side shows that very well. At different densities, you can see different morphological and behavioral changes in these cells, right? So on the left-hand side of the image, low density, the cells look and behave differently from the image on the right, which is high density. We see a lot more aggregating on the right. We see more dendritic-like processes being created by the cells. The morphology is changing. So just one simple example of how biologics are rarely a linear scale game. And this is also the case, to further highlight this, on the protein level. We can see, for example, that CHO cells transfected to produce an IgG isotype molecule do not behave consistently depending on your cell cultures. There is an increase or a differentiation in post-translational modification patterns of these proteins impacted by cell culture conditions. So in this case, looking at CHO cells fed glucose in different manners, either bolus fed or continuous fed, we see a resulting change in the post-translational modification patterns. Those CHO cells fed in a batch manner had greater heterogeneity in the post-translational modification patterns of these IgG isotypes, and it impacted both the PK and the ability of antibody-dependent cell cytotoxicity mechanisms. And what this group found was that feeding these cells in a bolus manner actually caused greater spikes in ammonia, which resulted in the increased heterogeneity of these post-translational modification patterns of the protein and the change in their efficacy and avidity and effectiveness. But by spiking these cultures with lactate, they were able to restore a more uniform pattern in the protein's post-translational modification patterns and ensuing functionality, as you saw when you continuously fed it. There are a lot of data on this slide, but you can see that there was a shift in alanine concentrations between the two cultures, as well as changes in viable cell concentration. And again, when they spiked these cultures with lactate, the profile was restored, the heterogeneity was decreased. So what you can see again is just another example of how scaling up on these different biological workflows is rarely a linear scale game. Now, let's shift gears on, let's talk about exosomes, define them a little bit, and then use them as a model to scale up and scale. Exosomes and EVs are not a new field of biologic, but they're renewed in interest. In fact, one might make the argument it's one of the original biologics, but I think we were challenged at the time of really understanding what we were looking at because size alone really is not a good single parameter for differentiating. In general, exosomes are between 30 and 150 nanometers in diameter, and this can vary. You'll pick up 4 different papers and get 4 different ranges, but somewhere between 130 to 150, maybe 40 to 200 nanometers. They tend to be exitosed out of the cells into the extracellular space. Now, this is in contrast to ectosomes, or shedding microvesicles. Now, as you see, they have an overlapping size range with exosomes, 100 to 1,000 nanometers, but they tend to really bud or bleb off of the cell membrane. They're not produced in their entirely internally and then exocytosed like exosomes are. Now, to further complicate this with respect to size range, right, we have apoptotic bodies, which of course are very different from ectosomes or exosomes, but you see here the potential size range of these apoptotic bodies as the cell goes through programmed cell death overlaps entirely with the exosomes and for the most part with the ectosomes. So again, size alone, while an important parameter and necessary, is not alone sufficient. And over the years, potential markers have been cited that could indicate exosomes and to differentiate them from, say, ectosomes or other subcellular biologics. Now, just so we're all sort of starting from the same level playing field, general considerations for EVs, specifically exosomes. Again, generally membrane-bound particles, somewhere between 30 and 150, 40, 200 nanometers. In vivo, found in virtually every cell type and fluid, and they are produced by cells in culture. Typically, they contain both proteins and nucleic acids, more specifically RNA. The proteins have been demonstrated to derive from both cellular sources, but there are also evolutionary conserved proteins, in most cases about 20,000 copies or molecules per exosome. In the case of the RNA, both messenger and silencing transcripts have been detected. They serve a myriad of functions, both in vivo and in vitro. In vivo, they're used as communication between biologics, right? Transferring materials to different cells, to coordinate behavior, to modulate production of proteins by affecting targeted genes, and to induce and coordinate immune responses. In the case of pathologies, right, many cancers have been shown to produce exosomes to confound or evade immune system function and or to hijack resources from the area to serve those cancer cells. So a myriad of different functions, and this is of course part of the challenge with identifying and characterizing these EVs. They're sharing these biologics, and so their profiles tend to be less static maybe than others. So again, there are a myriad of details with respect to functions, mechanisms, pathologies. We're going to focus on these exosomes for the remainder of the talk. Now, when we set forth to isolate and purify exosomes, there are a number of common techniques in the literature that I cite here. Differential ultracentrifugation. You may do some polymer-induced precipitation like PEG. Filtration, very common, of course. Antibody purification or affinity purification, typically by monoclonal antibody. And then density gradient centrifugation. Really, there are different benefits conferred or challenges faced by these techniques. So I don't know if there's necessarily always a one-size-fits-all, but it is often considered that differential centrifugation is the gold standard for exosome isolation and purification. This is something cited by Dr. Stephen Gould, who then cites these techniques and filtrations to isolate these particles here, these exosomes that you see down below. However, with this technique, there are a few things that are lacking, of course. There's no consideration, for example, from differentiating between, let's say, viral particle contamination or something like that. So those are the techniques. But before we really start using isolation purification techniques to obtain our exosomes of interest, we have to understand that there's a vast heterogeneity here in what we can choose. What I'm presenting is a publication from this group looking at the disparate isolation methods for extracellular vesicles and their impact on downstream RNA profiling. They cite 4 different techniques that they highlight. They either use ultracentrifugation, OptiPrep for their density gradient technique. They use a kit called ExoQuick and another one called Total Exosome Isolation. And they look at the different techniques and the ability to satisfy their workflow needs, and they summarize those in this table here. And they look at 4 things, right? So let's start with what we're focused on is purity and yield. And as you can see here, that while ultracentrifugation did do a great job with purity and yield compared to the isolation kits below, optical density gradient, OptiPrep centrifugation really did the best for purity and yield. Now, the picture's a little bit flipped here when you see the kits, while they did not purify and yield as many exosomes, the ensuing protein and RNA profiles were better with those kits than by centrifugation, either density or ultra. Nonetheless, for purity and for yield, density gradient was superior to all those techniques, and in fact, ultracentrifugation had the most balanced profile here. Good on purity, good on yields for the exosomes, the protein, and the RNA. But when it came to exosome purity and isolation, density gradient was the key, and here's their scoring matrix. Now, it is also important when we're working with these biologics is to control for contamination by exogenous sources. So let's start with this more simple protocol before we dive into this very, I think, complex yet extremely interesting publication that I'd like to present to you. When we're depleting EVs, centrifugation can play an effective role. And let's look at the example of media purification, in this case, fetal bovine serum, but also applicable to BSA as well. So we're looking here at a commercially depleted version of FBS for EVs versus a protocol to deplete these EVs in-house. And as a control, we're going to use the unadulterated source of FBS. And as you can see here, when we're tracking particle concentrations, here is our standard, the source, a fairly high concentration of exogenous EVs that could contaminate and complicate our downstream use in our EV research. Here you can see that the in-house depletion by ultracentrifugation was almost as effective as the commercially depleted version of the FBS. Now, to evaluate these three different conditions, what this group did was cultured cells in vitro for either 3 or 7 days and then counted viable cells by automated trypan blue exclusion and looked at viability. Now, day 3 in red, day 7 in the gray, and here in the unadulterated control source, we can see the nominal count of live cells. Interestingly, in the ultracentrifuge depleted FBS, we see a profile of cell viability very similar to the control. Now, interestingly, something about this commercially depleted version, the process by which they're pulling these EVs out, had some sort of effect in the cell culture. And by day 3, the viability counts were lower than the control or the ultracentrifuge. Now, by day 7, the cell counts, the viability was somewhat restored, although a little lower than the control and ultracentrifugation. But nonetheless, this commercially depleted version of FBS for EV did cause some variability in the predicted cell concentrations, the viability of these cultures compared to the ultracentrifuge. So one example, I think, of using ultracentrifugation very simply and effectively in controlling variability and maintaining consistency in your EV work. Now, there is a lot of heterogeneity in biological and biopharma protocols in general, and that stands true in the case of ultracentrifugation. And here we can see a technique where the two were combined with great efficacy. So isolating exosomes from Jurkat cells via ultracentrifugation, a multi-step process to remove cellular debris, to pellet for the exosomes. We used a biomech, an automated biomech, to set up density gradients so that we could scale up a little on the yields and the purity, and then performed further isolation and purification steps. So you can see here there can be a lot of different steps, combinations taken. It really depends on the biologics that you're specifically working with and the goals of the research and the protocols being used. Looking at factors that impact centrifugation workflows with exosomes, we have these 4 main factors here: viscosity, fluid density, the K factor, right, the efficiency of the rotor in the centrifugation system, time, and temperature. Now, what this study reference here by Momin Haravi found is that temperature and time really were not that impactful, and we're really going to focus on viscosity and K factor or in the rest of this discussion. So here I cite this calculation, this formula, to demonstrate viscosity of liquids. And here we can see that pre and post ultracentrifugation, so dark bars pre and lighter bars post, we're looking at different viscosities and their impact on isolating microvessels or extracellular vesicles from these different materials, whether it was plasmid, serum, culture media. And as you can see, pre and post prep, that this viscosity did have an impact on yield. So let's look a little further into this. Okay, so here are the data showing these concentrations pre and post ultracentrifugation, and then the differences in viscosity. What this study is really arguing is that the viscosity of the materials impacts the yield of exosomes or extracellular vesicles, and that really the lower viscous solutions yield greater concentrations of these microvesicles. Here is the equation for the sedimentation coefficient they used. And so this really boils down to also looking at K factor. So the time for a particle of sediment through a medium, and that's determined by the maximum angular velocity of the centrifuge and the min-max radius of the rotor. So RPMs alone are not a correct measure of the efficiencies of a centrifugation system. It is this calculation, the K factor, that is the true measure of efficiency. And again, what this group concluded was the viscosities of these different biofluids did affect EV isolation, the concentration, and that lower viscous biologics yielded more EVs. And what they also found was that a low angle fixed angle rotor provides better separation than a swinging bucket. So another parameter to consider when we're looking at isolation and purification of extracellular vesicles. So now let's look at centrifugation in the scale-up and scale-up processes. So as I showed you earlier, when we look at the literature, when we talk to our customers, when we consider our options for, you have these 5 techniques here again, right? We're going to ignore filtration for now, and we're going to ignore precipitation due to time constraints here. And instead, we're going to go again with what Dr. Gould had cited a number of years back, that differential centrifugation is the gold standard. Well, let's put that to the test a little more, huh? And let's take it a step further, and let's dive into this publication from Momin Haravi and colleagues, looking at the clinical utility of EVs and the fact that it will require standardized, efficient, automated methods with certified standards and controls in order for this have utility in the field. So again, there are a myriad of different sizes, surface markers, inner content, function, and physiology that overlap in these different subcellular particles. And so let's really look at differential centrifugation as a means of isolating these EVs. So in this case, we're going to compare differential density, in this case using iodixinol, versus an immunoaffinity pull down with antibodies. Now, when it came to density gradient by iodixinol, what they had used were CHO cell lines and compare those to plasmid samples in this process. They further characterized by these techniques protein content, microscopy, sizing, and proteomics. And here's the process, right? So they took these Lim863s, released the exosomes, collected also the plasmid, and as you can see here, sat these materials out at different temperatures for different amounts of time. And what this study really proved was that temperature maybe is not as impactful on the integrity of these EVs as we once thought. So diving into this process, looking at a couple of other cell lines as well, they isolated the extracellular vesicles using this protocol here, either through differential centrifugation and crude pelleting or density gradient by iodixinol. And comparing that to immunoaffinity pull down, here is what this group concluded, that in fact, the gold standard, once upon a time, alone was maybe a little bit of crude process for isolating these EVs from both the cell lines and the plasmid. Whereas the immunoaffinity pull down did a quite good job for the cell line and did a very poor job in the plasmid samples. And indeed, they found that density gradient separation had the best yields and purity in both of those sample types. So they then ran some gels on these different bands, these density gradients you see here from 1.04, 05, 06, 08, 10, 12, 14. And what you see here from these gels is that there were actually potentially different subsets or different expression patterns in these different density layers. And so at this point of the study, they decided to optimize the density gradient to increase the sensitivity, if you will, by looking at a greater number of fractions. And so you see here, they really kind of broke it down, particularly in the higher levels, to see if they could tease out some more distinction between what they saw in those gels. And as you can see here, as they optimized it, different expression patterns started to reveal themselves in these different cell lines, in particular these SKNBE3s, as you show here. And really what they started noticing was a further delineation when they improved the sensitivity of the density gradient. So from there, they decided to do some sizing studies looking at these different fractions. Here you see fraction 9, which is 1.08 all the way to 1.18 grams per milliliter, and you see different size subpopulations within this fraction. So it suggested to them, and you can see here from the imaging, that this was not a homogeneous single population, but perhaps a mix of several different populations of EV subtype. To further elucidate the difference in size in these potential subsets of EVs, visual studies were conducted via electron microscopy, and as you can see here in fraction 7, we have a subcellular particle in EV with this sort of morphological profile, right? Size is about anywhere around 80 nanometers. It looks from the size range, and when we compare that to the pelleted version here, you see a lot of variability in the size, a much larger size, and at least from my perspective, it looks like a cell membrane that maybe is under a little duress and showing some folding and some atypical profiles, perhaps due to the physical forces by density gradient. Now, compare that fraction 7 in the upper left-hand corner via density gradient to this fraction 910, and visually, clearly, we see two morphologically different populations here. The population here on the right seems to be of larger size. It's greater than 100 nanometers or more. We see maybe a little bit of a difference in the profile of the membrane. And again, to highlight that, if we show differential centrifugation down here, or dense ultracentrifugation rather, morphology. So indeed, by electron microscopy, we have now two different size populations by the Dulcimax, and here we can see by electron microscopy two morphologically different subpopulations. So to further elucidate what they were looking at, so what the group did is they looked at various RAB-GT paces. So these are intracellular switches that control the accessibility of these different EVs to the compartments of cells. And as you can see here, when we're looking at this exosome versus what they're now calling potentially ectosome group, certain RAB-GTPases are exclusively detected in exosomes, as shown here on the left. Conversely, these other ones here on the far right are exclusively detected in the ectosomes. So further showing differences in these internal switch and communication mechanisms. I think there's well over 60 of them associated with this process with extracellular vesicles. So again, some exclusively detected in the exosome population only, whereas others in the ectosome. And then in the middle, you see kind of a more heterogeneous, where some are expressed in both or slightly expressed in both. But nonetheless, two distinct RAB-GT paces become revealed when we look at these two different subtypes of EVs. Now, this to me is an extremely impressive summary slide. When you look at all of the work involved in monitoring these myriad of regulatory processes or biological processes here, from cell proliferation, metabolism, looking at the affected change in these biological processes in the presence of enriched or depleted exosomes and ectosomes. And as you can see, furthermore, in this depleted group, we have this huge decrease in regulation of translation, cell metabolism and proliferation, apoptosis, protein folding, and some other things. Where in contrast, in that enriched population, we see increases in immune system responses, cell-cell signaling, fatty acid metabolism, protein transport, and other signaling pathways. So definitely showing this distinct change in the biological processes as to whether the materials are enriched or depleted for exosomes and ectosomes. So really what this group, I think, showed was by optimizing their density gradient protocols, they were able to further isolate, purify, and then subsequently characterize potentially two distinct subtypes within this extracellular exosome subpopulation, right? We have this fraction 7, which are these small exosomes that are fairly consistent in size. Most of them fall within the 30 to 150 nanometer range. And as we saw earlier in the talk, they affect different changes in biological processes. They're of different morphology as well by electron microscopy. In contrast, this more dense fraction 9, these large EVs or ectosomes are more heterogeneous in size, although more than half of them are this larger 150 to 500 nanometer size. And they may be morphologically, physiologically, functionally distinct from this fraction 7, these smaller exosomes. And again, without them being able to use density gradient ultracentrifugation and really optimize their platform, their protocols to their needs, the study may not have been. So by taking advantage of the unique capabilities of density gradient ultracentrifugation to really further isolate and purify their subpopulations, which then allowed them to characterize them by size and functionality, from these results, using these 3 different cell lines, they proposed 2 distinct sets of markers to differentiate exosomes from ectosomes. And you can see these gels here in the middle, but on the left-hand side are the markers they proposed for exosomes, and on the right-hand side for ectosomes. Something, again, would not be possible without a technique that could really allow them to isolate and purify their target population of EVs. Now, one area, again, getting back to scale up and scale out, is in the production of these density gradient layers, right? So I think a lot of us are in the lab where there's one individual who's really good at layering these densities, and she or he happens to be on vacation for 2 weeks. What do? It can be challenging. So there are platforms, there are techniques where we can completely automate the density gradient layering, thereby helping address the throughput, right? The scale up component of scaling up and scaling out on extracellular vesicles. There really is no point to scaling up and scaling out until we have a well-defined, well-characterized target, and that we have a protocol that thoroughly isolates and separates the population of interest from those of non-interest. So again, until we know what our target is, it's very difficult to scale up and scale out. And as you saw earlier in the conversation, biologics is very a linear scale game. So if we want a technique that could really do a good job isolating and characterizing, the density layering gradient can be automated using our biomech automated system. And from there, we can perhaps propose a protocol that may get us towards where our goal is. And that is what Momin Haravi and colleagues said, until we have consistent, known, described processes with controls in place, it's going to be challenging to scale up and evolve EVs, exosomes, and other subcellular biologics. So here's an example of what this group proposed as a means of separating exosomes from ectosomes in our studies. So in summary, in centrifugation workflows for exosome isolation, much of what defines EVs and exosomes really still is in flux, right? We know size alone is necessary but not sufficient because of the overlap in size with other subcellular particles, and there really still is no consensus on what the definition of these particles are. So without basic characterization, other aspects are going to continue to remain poorly defined, I think. And so again, through a combination of ultracentrifugation, more specifically density separation, this is shown to be a highly effective means in EV scale up and scale out. We can work with larger amounts of high purity samples and have more consistent and predictable outcomes from our workflows, which I think is an important key to the next chapter in evolving the field of extracellular vesicles, exosomes, and now ectosomes. And so with that, I really would like to thank you for your time and attention. My contact information, my email is here below, and I'm happy to entertain any questions, comments, or discussions that the audience might like to have right now. So again, thank you.
And thank you, Dr. Lenz, for that informative presentation. We will now start the live Q&A portion of the webinar. And if you have any questions you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So it looks like we already have some great questions coming in from our audience. Derek, our first question is, EV isolation, its subtypes, is truly becoming too challenging, and though appear very important to cellular processes, hard to get traction in adopting EV research.
Yes, this is a very challenging topic, and I think, Rajesh, thank you for the comments and the questions. I don't disagree with you. When things are difficult like this, I like to be reminded of what my mentor said: if it was easy, it would be done already, and they wouldn't need us. So we have this challenge in front of us. And I think originally, as I mentioned, size alone was an insufficient parameter, and now we're seeing as more details become available, the picture becomes more challenging, right? Particularly since these biologic materials are being shared and transferred and effector functions enacted. So certainly it's going to be a summation of characteristics we're going to have to look at. Size, obviously, I think that the first logical place to look at is the evolutionarily conserved markers and proteins, and perhaps looking at something we can delineate akin to lymphocytes, right? Multiple markers, for example, to discern CD4 from CD8 T cells and other lymphocytes. It is difficult to get traction, and so I'm going to go back to the statement that we really, as a collective group, need to start deciding on standardized practices, controls, and known standards to get at this challenge. But I do share your pain there, Rajesh, and it's a good point to bring up. Nonetheless, there's a tremendous amount of potential clinical and application potential with these EVs. So I think it's certainly worth continuing to fight and get this figured out.
Thank you so much, Derek. Now, our next question is, given there's an overlap in size and density profiles for exosomes and other EVs, are there specific techniques to confirm the precise isolation and characterization of exosomes before their use in therapeutic development?
Yeah, that's Great question. I think that gets at the crux of another important aspect in this field. I think like most biologics, right, we have to take advantage of orthogonal techniques to verify what we're looking at, of course. So something like dynamic light scattering could be helpful for determining size profile. The challenge here, I think, Christy, and this is a good point you bring up, is again the overlap in size with these different subsets. And what that I think does sometimes is inadvertently gets us to the limits of sensitivity for certain techniques. For example, Coulter principle, right? A non-labeling direct manner of sizing particles like cells, very effective until we start getting down to around 220 nanometers. Then we have some challenges with resolution. I haven't seen a tremendous amount of research here yet, but I'm going to make a leap. And analytical ultracentrifugation, something that's been used very effectively for viral vectors and more recently lipid nanoparticles, could be a really good orthogonal technique for determining not only the size of the particles, but also the payload, the inner contents of the material. But as it stands right now, precise isolation and characterization, from what I can tell, is where many of us need to focus and really discern these methods before we can move on and scale up. But Christy, I'd be happy to talk with you offline if you wanted to have a conversation.
Thank you so much. Our next question, have exosomes been isolated from plants?
These are, by the way, this very engaged audience. Appreciate all the questions and comments. I like to hear talks, but this is my favorite part, right? Having a discussion and trying to educate one another on the state of the field. Yes, from what I can tell, and I'm not a botanist, but I have seen some publications where they are isolating, maybe not exosomes, but let's call them EVs from plants. So certainly there is the possibility that that biologic is present in that type of organism. Good question.
Thank you. And I also want to thank our audience for these great questions coming in. How about using ion exchange chromatography to enrich EVs before further ultracentrifuge isolation?
Yep, this is a common conversation. And again, I think it's very important to cite, we all want to find that silver bullet, that magic technique. I don't think that there is such a thing, right? So looking at these other techniques, such as ion exchange chromatography, it really depends what you want for trade-offs. So you can process high volumes, of course, with chromatography, but the challenges I've had conveyed to me from customers and other researchers, friends of mine in the field, is that you can lose a lot of the yield in the clumping in these columns. And so you'll be able to trade off, right, process high volumes. It's just that the yields, the ROI may not be sufficient for your downstream needs. So it's important to look at these different techniques and to understand the trade-offs and determine what's going to be best for your specific needs. So if I was looking for high purity, high yield, ion exchange chromatography might be counterproductive. But if I had huge volumes and that I wanted to process quickly, I might keep that as an option. Good question.
Thank you, Derek. Now, Derek, do exosomes and other EV play a role in disease pathogenesis, or are they merely a marker of disease?
Christy, you are asking great questions today. All of you are. Yes, there does seem to be the case in both instances. Now, let's talk about the disease pathogenesis, specifically cancer. We have observed extracellular vesicles being released by the cancer cell to impact signaling and local biogenesis. We've seen examples of materials being released so that the cancer cells can hijack the vasculature, right? Get a greater blood flow, oxygen flow, and more nutrients to it. We've seen examples of cancer cells releasing EVs to evade and confound the immune system. But also, to your point, in other cases, we can see these EVs as indicators for disease. So I think the literature points to to both these situations, and it really just depends on the disease indicator of interest.
Thank you. Now, Derek, how does this harvesting exosome pave way to cancer research?
Yep, great question, John, and good timing, right? Let's follow up on that. If we are able to have more consistent, predictable techniques for isolation and purification, then obviously by logic that impacts positively all your downstream research. It could be the case, hypothetical here, that we may be looking at two different distinct populations of exosomes, and one may be cancer derived and one may be non-cancer derived, or they may be two different subsets of exosomes or EVs coming from the cancer cells that ultimately affect different functions. But I think really, I bait and switched this talk a little, I'll admit, right? We wanted to talk about scale up and scale out, but the point is, since biologics is rarely a linear scale game, we really need to double down on our isolation and purification before we take that next big step. I hope that answers your question. If it doesn't, please let me know and we can continue the conversation offline.
Now, we have time for a few more questions, Derek. Do you think it's possible to isolate exosomes from exosomes based on ultrafiltration steps downstream of chromatography?
Certainly possible. And thank you, Fatima, for the question. The concern here, I think, when I look at the data in the literature is the physical forces and their ensuing negative impact on the morphological profile of our target biologic. So I think you saw in the case of the pictures from electron microscopy that the differential centrifugation compared to density gradient, the materials looked a little different, right? Again, and from my take, the density gradient EVs looked a little more true to expectation, whereas when we subjected it to differential and those high physical forces, centrifugal forces and G forces, it seemed like the cell lost, or the material lost a little bit of its integrity. So my concern here is we do chromatography, we may lose a lot of our materials, and then we subject it to ultrafiltration. The question becomes how much and what are the quality of what you have left over? The literature suggests to me instead that we optimize density gradient centrifugation and perhaps understand the markers associated with those targets and then move in another direction. Maybe we do density gradient and then from there, immunoaffinity pull down, for example. But great question.
Thank you so much. Now, you mentioned K factor for centrifugation for efficiency or something similar. Could you review that a little bit more?
Yeah, absolutely. This comes up quite a bit. Now, once upon a time, as a young scientist, I was guilty of this as well, right? We have too much on our plate, and we have to know a little bit about everything. I think there is an assumed mistake when we're evaluating our centrifugation protocols that the RPMs are alone the single parameter for deciding the efficiencies when that is indeed not the case. The true measure of efficiency in a centrifugation protocol is the K factor. And the K factor takes a couple of variables into consideration. Now, it doesn't take temperature into consideration, but it's the maximum speed as well as the path length that the particle travels within the tube or the container it's being held in. And so those factors together equal the K factor. Generally, what it suggests is the lower the K factor, the shorter the runtime. So RPM alone is not sufficient. We have to consider the path length that the particle travels, and those factors together are referred to as K factor, and that is the true measure of efficiency. But great question. Always happy to have that discussion.
Thank you so much. Now, our next audience member says, we're having some trouble with consistently determining content of our exosome particles. Could you suggest an analytical technique for particle payload?
Yep, this is a good question, and I think something that we're always kind of wrestling with in the back of our research minds. There are other orthogonal techniques we can look at. For example, we could use dynamic light scattering to further separate by size. But when it comes to that particle payload, there is not a lot of literature out there yet. But I would be surprised if the technique of analytical ultracentrifugation can do for extracellular vesicles what it's been doing for viral vectors and more recently for lipid nanoparticles, where we're using separation plus interrogation either by UV-vis spectrometry or in parallel with Rayleigh interference. We can really discern how much content is inside that particle by its separation techniques. You can see this with genomic payload for AAV in gene therapy, for example. So in my mind, analytical ultracentrifugation could be an extremely powerful technique, not only for that parameter, but also for size and ultimately other orthogonal techniques. So good question.
Thank you, Derek. We have time for one more question. Our audience member says, if I were to use flow cytometry for EV analyses, would you also recommend adding tetraspanin CD63? And second part of the question, also, if I wanted to analyze internal contents, could you recommend affixing and staining protocols?
Wow. Okay. So there's a lot in this question. Let me see if I can get through it in the time left. First of all, we have to be cautious. I'll cite earlier what I said, the limits of resolution for size on some of these techniques. Look at the flow cytometer that you're using, right? Some of them, Beckman, for example, can look fairly well below cellular, but will not be able to measure the full range of sizes of potential for EV. So that's one challenge. From what I can read, from what I see, right, the tetraspanin is typically kind of a pan-EV marker. And so again, I think what's going to happen ultimately is something akin to what we see with lymphocyte analysis, right? Something I'm extremely familiar with. Looking at multiple markers to distinguish maybe CD4s from CD8s, and then memory CD4 from naive, activated, so on and so forth. As far as a fixing and staining protocol, that's a tough one. I've had a lot of experience there. In some cases, fixing with paraformaldehyde and doing proliferation assays on cells is no problem. But before I answer that question, I'm going to have to look into the literature a little bit more, because there's a lot of variability in the lipid profiles of these different particles as well. So I wish I could answer that in its entirety, but I don't want to provide anything that I'm not 100% certain on.
Derek, I want to thank you again for your time today and for your important research. Would you like to provide any closing remarks before we go?
Absolutely. I'd like to thank your group to start off with. You're absolutely wonderful to work with. Thank you to the audience. I hope it was time well spent. Seemed very engaged. We appreciate all the questions. And please, if you want to continue any conversations or have further discussions, do not hesitate to reach out. But thanks, and enjoy the rest of your day.
And thank you, sir, for your presentation. And we would also like to thank LabRoots and our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. And before we go, I want to thank our audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speaker via contact information you provided at the time of registration. And this webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone, and bye-bye.